All right, this video is going to talk more about the nitty-gritty details of DNA replication. So in your microbial genetics PowerPoint, I list several key players that are involved in this. And so I'm going to talk about them a little bit as we go through this animation, okay? So this particular slide and animation is going to focus on the key players that are more involved in actually creating the new strand of nucleotides. But I do want to remind you that originally, right, this replication fork was closed, right? The two strands were completely linear. And it was helicase's job to separate those two strands. It helps break those hydrogen bonds that's connecting the two parent strands. But remember, those two strands of nucleotides like being together. And so they kind of naturally want to reattach. And so we have single-stranded binding proteins, which I always call SSBPs for short. Their job is to quickly attach to the newly separated parent strands and keep them attached. So I visualize them as like bumpers to prevent the two parent strands from reattaching to each other. So those single-stranded binding proteins keep these strands apart so we can have the replication fork that you see on the screen. Now, the other enzyme that you can't see at the moment is topoisomerase. And that one works way down at the end, at the piece of the DNA or the region of DNA that's still closed. And remember, its purpose is to relieve supercoiling. Because as a helix is literally, the two strands are separated, it causes the rest of the existing helix to get really tightly wound up. And that can cause the DNA to break, which obviously would be really bad. So topoisomerase's job is to keep that from happening. It keeps the helix in a nice, relaxed coil. Okay, so now we're going to talk about the other enzymes or key players that are going to actually be creating the new strand of DNA. I'm sorry, the new strand of nucleotides. But remember, the new strand cannot be made without helicase, single-stranded binding proteins, and topoisomerase first and continuously doing their job. Okay, so as the table in your PowerPoint says, we have two DNA polymerases. And remember, enzymes are named with ASE at the end. So if you kind of cover up the ACE, because that's just telling you that the molecule you're talking about is an enzyme, the rest of the name is normally a hint as to what it does, right? So think back to helicase. If you ignore the ACE, it's helic, which sounds a lot like helix, and its job was to separate the helix, right? Separate the strands of that helix. So the name is a clue as to what's going on. So DNA polymerase, if you remove that ACE, you have DNA polymer. So that means this enzyme is making the polymer known as DNA. Well, in order to make DNA, you have to lay down nucleotides, right? Remember the last video where it showed each base was being added one at a time? That's what DNA polymerases do. Now, DNA polymerase 3 is kind of the star of the show. It does most of the nucleotide incorporation. But I think of it, how I remembered it when I was in school, so now how I teach it, is I think of DNA polymerase 1 and 3 as like being a family. There's a DNA polymerase 2 out there, but we don't care about that one. So I think of DNA polymerase 1 as the firstborn, so like the big sister. And I think of DNA polymerase 3 as the thirdborn, and in my story, that's the baby of the family. Right? So in my story, the baby of the family kind of gets what it wants, right? It gets to be a little high maintenance. And the firstborn just kind of has to deal with it, right? Kind of clean up its mess. So, well, first of all, no offense to anybody that's a firstborn or a baby of the family. Just a story to help you remember. Okay, So DNA polymerase 3, being the star of the show, being the baby, he kind of has some demands in order to do his job, or even some limitations. So it can only add nucleotides to a free 3' end, and this will make more sense in a second, and it cannot start from scratch. So that means DNA polymerase can't just look at this open, free region of DNA and start creating the new strand off of it. It needs help getting started. Okay, so let's visualize this. So right now we're only going to look at the top orange strand, and that's actually called the leading strand. Okay, the bottom strand is called the lagging strand, and you'll understand why later. So we're going to get the leading strand. So remember, DNA polymerase 3, high maintenance, cannot start from scratch, so it needs some help. So the enzyme primase is going to be the enzyme that actually starts the process of making that new strand of nucleotides. And so primase, again, get rid of the ace, says prime, and that's what it's doing. It's priming the process. And it does that by laying down a primer, okay? But it is an RNA primer. So what that means is primase lays down a small region of RNA nucleotides. Okay, remember, RNA nucleotides are a little different than DNA nucleotides, okay? So this is going to be a little problematic later, but nothing the cell can't handle. So this little piece of RNA is laid down by primase. Now, RNA is also a nucleic acid, just like DNA. And so this piece of RNA that's laid down is going to be anti-parallel and complementary to that parent DNA strand, okay? So notice the directionality is there. And I put that because remember, not only did DNA polymerase 3 need help getting started, but it can only add DNA nucleotides to a free 3' end. And that's what we have, right? Do you see where the 3' is on that primer, and how there's plenty of DNA on that top strand that can serve as a template? So DNA polymerase 3 is like, okay, I guess I'll do my job. And so it comes in and it starts adding DNA nucleotides. And it just keeps on going. As long as there's a template, like a parent template strand to use, it just creates that new strand of DNA. Okay, so we're going to stop there at the fork because remember that other region is closed. And let's address this RNA primer because we can't have RNA mixed in with our DNA. And so what's going to happen is this is where DNA polymerase 1, the big sister, comes in and kind of cleans up DNA polymerase 3's mess. So DNA polymerase 1 has two functions. The first is to remove the RNA primer. But that leaves us with another problem. Now there's a region of that top parent template strand that has not been used, right? We're missing part of our newly made nucleotide strand, right? That blue strand is not complete. So the second job that DNA polymerase 1 has is to fill in that gap. And that's what it does. But notice I left a gap between the small piece that DNA polymerase 1 added and the long chain of nucleotides that DNA polymerase 3 had already laid down. And that's because everything has its limitation. And DNA polymerase 1 cannot attach the little strand it made to the existing strand made by DNA polymerase. It needs help linking those two pieces, okay? And so that's where our final enzyme comes in, ligase. Ligase means ligate, and ligate means to connect. So that enzyme's sole purpose is to come in here and connect the strand laid down by DNA polymerase 1 to the strand laid down by DNA polymerase 3. So now we have a nice completed strand of DNA nucleotides. Now, at this point, what would happen? Okay, we're going to talk about what's going on the bottom strand. It's the same process, but a little more complicated. But once this open region of the fork has been used as a template to create that new strand, then this currently closed region, right, on the left would open up so that we could then use it as a template to keep the new strand going. But let's talk about the lagging strand. It's a little bit more complicated, but again, the exact same steps. So DNA polymerase 3, the baby of the family, needs to come and create this new strand of DNA nucleotides. But remember, it cannot start from scratch. So primase jumps in and lays down a small RNA primer. But here's the problem. Because, at least in this picture, the bottom strand runs 3' to 5', which is opposite of that top strand, look at the directionality of the RNA primer. Do you see how it's running opposite? So it's 5' to 3'? Well, that's a problem because DNA polymerase 3 can only add nucleotides to a 3' end. And there is no free nucleotides on the 3' end, right? That's the very edge of the fork. So what actually has to happen is primase has to go and add another primer a little farther up the fork. So there we go. Primase laid down another RNA primer. Again, still in the 5' to 3' direction, but that's okay because now there is a free 3' end that DNA polymerase 3 can use. So then DNA polymerase 3 fills in that section, okay? But now you might be thinking, what about the rest of the fork? Well, primase really has to put in work on this lagging strand. So oops, I got ahead of myself. Primase is going to lay down another piece of primer, again, piece of RNA nucleotides, and DNA polymerase 3 will fill in the gap. This is going to continue away, okay? But remember, we cannot have RNA nucleotides in our DNA. So who comes and removes them? The big sister, DNA polymerase 1. So DNA polymerase 1 is going to remove the primer, and I removed the one at the 5' end of that orange strand. And DNA polymerase 1 is then going to fill in the gap. But remember, DNA polymerase 1 cannot completely connect to the existing strand that DNA polymerase 3 had created. So there's that little gap. So which enzyme comes in and fills in the gap? Ligase, because remember, ligate means to connect. So it connects the two strands. All right, well now, dpol, that's shorthand for DNA polymerase. DNA polymerase 1 is now going to remove the next piece of RNA primer, fill in the gap. Ligase will connect the strands. DNA polymerase 1, again, get rid of the primer, replace it, and then ligase connected them. So now I'm just trying to kind of speed up the process. DNA polymerase 1 replaces the gets rid of the primer, replaces the DNA nucleotides, and ligase fills it in. Okay, and this happens the entire way. Okay, so when we're talking about making the new strand of DNA, primase comes first, which allows DNA polymerase 3 to then start adding the new DNA nucleotides. Okay, and on the lagging strand, this happens in pieces or fragments. And so those individual fragments are called Okazaki fragments. Let me see if I can spell that from memory. Okazaki, Okazaki fragments. And these are only on that lagging strand, okay? The leading strand is made continuously. Once there's a primer, DNA polymerase 3 can just chug away, okay, because of the directionality of that original top parent strand. But on the lagging strand, it's in pieces. So primase keeps laying down bits of RNA primer. DNA polymerase 3 will fill in those available regions. DNA polymerase 1 will then remove all of those primers, fill in the gaps with DNA nucleotides, and then ligase will kind of finish up the process by connecting all of those strands together. So we have one continuous piece or strand of DNA. Okay, now again, once the open region of the replication fork has been used completely, then the rest of the fork will open up and this same process continues until we have two complete daughter molecules of DNA. All right, y'all, that is going to be the end of this video. Let me know if you have any questions.